Hello students, welcome to Digital Learning DCE. Myself Dr. Manjula, working as Assistant Professor of Botany in Government First Grade College, Ramanagara. In this session, let us study about general characters of fungi with special reference to occurrence and thallus organization. Let me begin with Introduction to Mycology. Mycology is derived from two words. Myx means mushroom, logos means study. So it is the branch of botany which generally deals with study of fungi. Fungi is a plural word of fungus which is derived from Latin word fungar, which means to flourish. This word fungi is used to designate mushrooms, hence the name fungi. So mycology is the branch of botany which generally deals with study of fungi. So let me define what are fungi. Fungi are either unicellular or multicellular, eukaryotic, achlorophyllous and heterotrophic thallophytes. Eukaryotic means the organisms which have well developed nucleus along with membrane bound organelles like ribosomes, endoplasmic reticulum, Golgi apparatus, mitochondria, etc. Achlorophyllous means since the fungi lack chlorophyll, they are called as achlorophyllous organisms. So they cannot synthesize their own food. They have to depend upon another organism for getting food. Hence they are heterotrophic in nature. Coming to thallophytes, thallophytes means the organism where the plant body cannot be distinguished into root, stem and leaf. Those organisms are referred as thallus. In case of fungi, the plant body consists of thallus, which is made up of a thin filamentous thread-like structure called hyphae. The hyphae interwoven together to constitute a network-like structure called mycelium. So let me give some examples to fungi which we are seeing daily life. Yeast, which are generally involved in fermentation and also in bakery. Certain molds like bread mold, black mold, etc. And mushrooms. Some of the mushrooms are edible, and some puffballs which are going to be developed where the organic matter is very rich, and certain other fungi like rust and smut which are going to cause diseases in plants. Till today, about 5,100 genera and more than 50,000 species of fungi are known. So in this slide we can see some pictures of fungi. This is a yeast cells and this is puffballs and these are mushrooms. Coming to general characters of fungi. First one, occurrence. That means the distribution. Where these fungi are distributed or occur in nature means they are cosmopolitan in distribution, otherwise known as ubiquitous in distribution. That means they occur everywhere where life can be possible. They found as saprophytes, parasites or symbionts. Coming to the plant body, that is referred to as thallus. The thallus of fungi is made up of with either unicellular or multicellular. Unicellular means the fungal cell, the whole plant body is made up of with a single cell. Example, yeast or Saccharomyces. Coming to the multicellular organisms, the thallus is made up of with many cells. In that case, multicellular, the filaments are present, thin thread-like filamentous-like structures are present. Those are referred to as hyphae. These hyphae interwoven together to form a net-like structure called mycelium. The hyphae may be, in case of multicellular organisms, the hyphae may be either septate or aseptate. Septate means the hyphae, thin thread-like structure, is provided with the cross walls. Then it is said to be septate hyphae. If the hyphae lacks cross wall, then it is said to be aseptate. So in this diagram, we can observe both septate and aseptate hyphae which are multicellular in nature. Then coming to the septate hyphae, as I already told, septa means the cross wall. In case of septate hyphae, Either they may be uninucleate, binucleate or multinucleate. And this septa cross wall, they are provided with a small opening called pore. So if the pore is very simple, then that is said to be simple septum. If the pore is provided with certain dome-like structure, then that is said to be dolipore septum, which is the characteristic feature of Deuteromycetes fungus. Coming to the aseptate type A, since there is no cross wall in this hyphae, always the nucleus are more in number. They are freely floating inside the hyphae. So that condition is said to be cenocytic. I mean to say, the multinucleate condition of aseptate hyphae is cenocytic in nature. Next, coming to the hyphae, are either homo or heterokaryotic in nature. Homokaryotic hyphae means all the nucleus are of similar strain, whereas in case of heterokaryotic hyphae, the nuclei is from different strains, and the hyphae may be either haploid or diploid. Haploid phase is exhibited by asexual reproduction of the fungi, whereas the diploid phase is exhibited during the sexual reproduction of the fungi. Then coming to the cell structure, I already told it is either unicellular or multicellular, and the cells are eukaryotic in nature. Eukaryotic means they are the organisms which are having advanced nucleus made up of with well defined nucleolar membrane and also nucleolus. And along with that, there are certain other membrane bound organelles like ribosomes, mitochondria, endoplasmic reticulum. All those are floating inside the cytoplasm. Those type of organisms are called as eukaryotic organisms. So the cell structure of fungi is eukaryotic in nature, and the outermost layer is called the cell wall. The cell wall is made up of cellulose. In case of fungi, there is a special type of cellulose called chitin is present. And next to the cell wall, there is a membrane, thin membrane, that is called as either plasma membrane or cell membrane, which is enclosing internally a liquid called, colorless liquid called cytoplasm. The cytoplasm in turn encloses the nucleus and other membrane bound organelles. The nucleus status is eukaryotic, which is floating, nucleus is floating inside the colorless vacuolated cytoplasm along with certain organelles like endoplasmic reticulum, mitochondria, Golgi apparatus, ribosomes, etc. Then, in case of higher fungi, the motile cells are absent. Motile cells means the cells which are provided with locomotory organs. But in lower fungi, the reproductive cells like zoospores are provided with one or two flagella. Flagella means they are the air-like structure whose to and fro movement bring about the locomotion in organisms. The flagella in case of fungi may be of two types, whiplash or tensile. Whiplash flagella is a thin, long, hair-like structure, whereas in case of tensile, this thin filament is provided with small hair-like outgrowth on its surface. In most of the fungi, hyphae form a felt-like cottony structure, but in advanced fungi, the hyphae undergo certain modifications in response to their functional needs. So such modifications includes Prosynchyma, Rhizomar, Sclerotium, Aprosorium, Astoria, Eiffel Traps and snares, and lastly Stroma. Let us study all these modifications one by one in detail. The first example of Eiffel modification is Prosynchyma. Prosynchyma is a loosely interwoven Eiffel tissue which lie almost parallel to each other. So in this diagram we can observe number of hyphae are arranged one nearby another one, which are parallel in nature, and they can be easily distinguishable. They are loosely interwoven Eiffel structure. So they generally modify to give support, and sometimes they also helps in conduction from one cell to another cell. Then coming to the second example in Eiffel modification, that is Pseudo Parenchyma. Pseudo Parenchyma is a compact massive parenchyma-like tissue. Why it is called Pseudo Parenchyma means not exactly it is a parenchyma cell, but it looked like a parenchyma. Hence the name pseudo, false parenchyma. So here the hyphae are compactly packed and they are very closely packed and interwoven. In the cross section, these hyphae appear like isodiametric cells where hyphae are going to lose their individuality and they are not distinguishable from one another. So this modification is mainly for storage of food material. Then coming to the third type of hyphal modification, that is rhizomorph. As the name itself indicates, rhizo means root, morph means external structure. So in its external structure, rhizomorph look like a root of a plant. So in this case, several hyphae become interwoven with each other and forms a thick strand or root-like aggregations called rhizomorph. In the diagram, we can observe some root-like structure which are the modifications of fungal hyphae. They mainly helps in penetration, increase the infection capacity of hyphae. So these modifications for penetrating the host. And coming to the next example of hyphal modification, that is sclerotium. It is a compact globose structure formed by the aggregation and adhesion of hyphae, where the outer hyphae become very hard and it forms a protective covering to the inner hyphae. They remain dormant, that means they won't interact to external environmental condition. So they survive for longer periods and thus it represents the resting stage of the fungus. They accumulate reserve food materials and they generally helps in vegetative propagation. They act as a vegetative propagule units. On return of favorable condition, the sclerotium germinate to produce new hyphal cells. Coming to the next type of hyphal modification, that is Aprosorium and Astoria. Aprosorium and Astoria are special hyphal modifications usually observed in case of some parasitic fungi. The Aprosorium is a terminal simple or lobed swollen structure, so which is formed at the tip of the germ tube. In case of parasitic fungi, when the spore fall on the substratum or on the host, they are going to produce a tube called germ tube, and this germ tube at its tip it is going to bulge and form a lobe-like structure. This is referred as Aprosorium, but is filled with certain enzymes, and these enzymes dissolves the wall layer of the host so that it is easy to enter the germ tube into the host tissue. So Aprosorium helps in penetration of the host tissue. Then coming to the next hyphal modification, that is Astoria. Astoria are the finger-like projections inside the cell, which are generally referred as absorbing organs. They are present in parasitic fungi where they are either finger-like, knob-like, elongated, branched structure. They are going to secrete some enzymes which hydrolyze the protein and carbohydrates of the host cell and getting their nutrition. So usually they are intracellular absorbing organs. Then coming to the next hyphal modification, that is Eiffel traps or Eiffel snails. So these structures are usually observed in case of predaceous fungi, which develop sticky hyphae or network of hyphal loops known as Eiffel traps, which generally helps in capturing nematodes. Then the last type of hyphal modification is stroma. Stroma is a thick black mycelial mat on which large number of fructifications are going to be formed. So these fructifications are nothing but fruiting bodies of the fungi which are going to enclose number of spores for the next generation. Then coming to continue with the general characters, that is nutrition in fungi. As I already told, since fungi lacks chloroplast, they are achlorophyllous in nature. They are unable to synthesize their own food. They have to depend upon another organism for getting the food. Hence they are called as heterotrophs. So heterotrophic mode of nutrition is of three types, either parasitic, saprophytic or symbiotic. In case of parasites, these are the organisms which are going to obtain their food from other living plants or animals. The parasites may be obligate or facultative. Example for parasitic mode of nutrition observed in case of fungi is Puccinia, Ustilago and Specilotheca. Coming to the second type in heterotrophic mode of nutrition is saprophytes. Saprophytes are the organisms which are going to obtain their food from dead and decaying organic matter. Example, mucor and pythium. The third category is symbionts. Symbionts are the organisms of fungi which grow along with other living organisms where both of them are mutually getting benefited without affecting one another. Example of such mode of nutrition is carried out by lichens and mycorrhiza. So in the diagram we can observe the parasitic fungi which is infecting the plant and getting their nutrition through Hostoria. These are the saprophytic fungi which are usually growing in dead and decaying organic matter, and this is an example of lichen which is an association between the fungi and algae. Then coming to the types of parasitic nutrition. Parasitic fungi are of two types, obligate parasite and facultative parasite. Obligate means they are strictly require the living host. Without the host, they won't exist. Their growth is not possible. So those type of fungi are called as obligate parasite. Example, Puccinia. Then the second type in parasitic fungi is facultative saprophytes. So here, these are actually the parasitic fungi, but in the absence of their host, they also have the capacity to grow on dead organic matter. So such type of fungi examples includes Specilotheca and Ustilago. Coming to the types in saprophytic fungi, first one is obligate saprophyte. So these obligate saprophyte obtain the nutrition only from the dead organic matter, but they are not able to infect the plant or animal. Even though get their host, they are unable to infect them. So these are called strictly saprophytes, obligate saprophytes. Then the second category is facultative parasites. These are actually saprophytic fungi, but if they get the chance to infect the host, they have the capacity to infect living organisms. Hence they are called as facultative parasites. Example, Fusarium and Pythium. So in this picture we can observe the Puccinia infecting barberry plant. Then coming to the second one, this is actually Specilotheca. It is a parasitic fungi. In the absence of its respective host, it can also grow as a saprophytic fungi. And this is an example for obligate saprophyte, that is mucor. Mucor is generally called as the black mold, which is developed on the substances which are rich in organic matter. And in the fourth picture we can observe the facultative parasite. That means it is actually a saprophytic fungi. When it gets the host, it has the capacity to infect that. This is a Fusarium wilt caused by Fusarium in tomato plant. Then coming to reproduction in case of fungi. Fungi reproduce by three important methods like vegetative method, asexual method and sexual method. So let us discuss one by one in detail. Vegetative method, vegetative reproduction in case of fungi takes place by three important methods like fragmentation, fission and budding. So in case of fragmentation, this is a filamentous hyphae, multicellular hyphae, where if there is any mechanical injury to the filament get broken, and this broken filament has the capacity to develop into new individual hyphae. This type of reproduction is called fragmentation. And the second category under vegetative reproduction is budding. So here a small outgrowth called bud is going to be developed, into which a small amount of nucleus along with bit of cytoplasm enter, and there is a constriction develops, and at the point of constriction this bud detaches from the mother cell and it has the capacity to lead into individual hyphae, which is usually observed in case of yeast. And coming to the fission, so it is usually nothing but dividing cell. So the mother cell, it is going to divide equal proportion of cytoplasm and nucleus, and a constriction appears, and this constriction extended and this gets separated into two daughter cells which has the capacity to develop into new cells. Coming to asexual reproduction, so majority of the fungi till today identified fungi, 20% of the fungi propagate by this method. So which is usually by the formation of conidia and other various spores like zoospores, chlamydospores, idea and applanospores. We are going to study this asexual spores in detail in the next session. And this asexual spores are either unicellular or multicellular. They may be endogenous, that means they are produced inside the sporangium or pycnilium, or exogenous. If they produce towards outside on a special branches called conidiophore, then in that case they are exogenous in nature. If they are produced inside a sporangium, then in that case these asexual spores are endogenous in nature. Then coming to the sexual reproduction, except class Deuteromycetes, which is otherwise known as imperfect fungi, all the class fungi reproduce sexually. The sexual reproduction takes place by three important methods like plasmogamy, karyogamy and meiosis. Plasmogamy is nothing but the fusion of protoplasm, which is followed by the second phenomena called karyogamy. Karyon means nucleus, so gamy means fusion. So here the nucleus of different strain, nuclei of different hyphae is going to mix up together and form a diploid zygote. This diploid zygote undergo meiosis, which is otherwise known as reductional division, to form again haploid spores. So in this diagram we can observe the sexual reproduction taking place in fungi where the two strains of opposite strain, two hyphae of opposite strain come close together. First phenomenon is plasmogamy, that is mixing up of the protoplasm, and then it is followed by, so that it results in the dikaryotic mycelium where two nuclei are present inside a single cell. Out of two, one is from male gamete and another one is from female gamete, and this two nuclei is going to fuse to form a diploid zygote. This zygote, that phenomenon is called karyogamy, and after karyogamy this diploid nuclei or zygote undergo meiotic division to form again haploid spores. So the life cycle continues. Coming to the life cycle of fungi, they have distinct haploid and diploid phase. So fungi reproduce asexually by haploid phase, whereas sexually by diploid phase. So always haploid phase alternate with the diploid phase. Hence fungi exhibit alternation of generation in their life cycle. So with this, let me give summary of this session, that is general characters of fungi. Mycology is the branch of botany, generally deals with study of fungi. The fungi are cosmopolitan in distribution, that means they occur everywhere where life can possible, either as saprophytes, parasites or symbionts. The plant body is referred to as thallus, which is made up of branched hyphae called mycelium. Hyphae may be thin thread-like structures called filamentous structures. They are provided with cross wall, then it is said to be septate. If they are not with cross wall, then they are aseptate in nature. And hyphae in higher forms modified into various structures like Prosynchyma, Rhizomar, Sclerosium, Aprosorium, Astorium, Eiffel Traps and Stroma. And the fungi are eukaryotic in nature with advanced nucleus and other membrane bound structures like endoplasmic reticulum, mitochondria, Golgi apparatus, but lacks chloroplast. Hence they have to depend upon another organism for getting their nutrition. Heterotrophic in nature, either by parasitic mode, saprophytic mode or symbiotic mode, and they reproduce by vegetative, asexual and sexual means the fungi exhibit alternation of generation in their life cycle with haploid and diploid phase alternating each other. So with this I am going to conclude this session. For further details about the topic you can refer the following references. Thank you.